Begin now. Good morning, everybody, and welcome to Grand Rounds. This will be final Grand Rounds, actually, of the calendar year 2021. For the next 2 weeks, we'll recess because of the holidays and resume on January 5th when there will be a clinical update in psychiatry Grand Rounds, which will include Jeremy Kidd on developing psychotherapeutic approaches for LGBTQ populations, Arthur Williams on telehealth for medications for opioid use disorder, and Samantha Lookatch on the craft approach for treating addiction. I was mentioning and talking with our speaker and moderator beforehand that unfortunately COVID is on the rise again, nowhere near as bad as it was in the spring of 2020, but still the numbers are going up rather than down. So the same precautions that we've been taking need to be maintained in terms of masks, social distancing, ensuring vaccination, testing when you need it. And in the ground floor of the Pardis building, where the mailroom used to be, there is a rapid testing site that you can go into ad lib and have a test anytime you want. Well, it's a real pleasure to introduce Dan Salzman today. Dan is one of our really unique and most distinguished faculties in terms of his expertise and his background. Back when I was training, the field of psychiatry was just beginning to avail itself of the powerful and sophisticated technologies of basic science. And although you did have some people like the Eric Kandells and the Solomon Snyders and the what was Serenello's first name? Roland or Dominic? Roland Serenello, was it, Dan?
Roland, yeah, I think he's the one who tragically died at ACMP. Am I right?
Exactly. But there were individuals that were really trained in basic science disciplines. But it was still somewhat remote from mainstream scientific psychiatry and psychiatric medicine. And thankfully, that gap has been steadily closed, as exemplified by our speaker today and also our moderator today, Christine Denny. But Dan took a particularly challenging road to achieve his professional aspirations. But to begin, he was born actually in New York, Staten Island. He went to undergraduate school at the University of Massachusetts in Amherst, and then he went west to Stanford, where he received a medical degree and a PhD working in the laboratory of Bill Newsom, who was a distinguished neurologist and neuroscientist working in subhuman primates. And Dan did his thesis on neural mechanisms underlying judgments on motion direction. Following graduation, he was on faculty briefly before he was recruited in 2001 to Columbia, where he was recruited into the Department of Neuroscience, but also an appointment in the Department of Psychiatry, and resided in the Cole building, where the Center of Neurobiology and Behavior with Eric Kandel and company resided. Dan has risen in terms of the academic ranks to a full professor in neuroscience and psychiatry with tenure, and has run a very successful laboratory which is focused on studying basic mental functions and their relationship to mental disorders using subhuman primate and rodent models. And he's taken advantage of his expertise as a psychiatrist and neuroscientist. He succeeded Josh Gordon as the director of our Leon Levy Fellowship Program, and also is the co-director of the T32 that our department has in research training in affective and related disorders. In the context of his work, he's been an active teacher and a generative mentor with various types of graduate students and postdoctoral fellows, including some of our own graduates, including Sylvia Bernardi, Lee Lovejoy, and currently Alejandro Ramirez. The work that he's done, and why I said more challenging, is because primate research is a a limited area in terms of truly expert researchers, and it is in many respects a more challenging area to master. It takes a long time to develop the expertise, and the performance of studies, of experiments also can be prolonged. And in this case, Dan's really a true example of the less is more, in the sense that he's a full professor in 2 disciplines, and he's achieved great accolades and visibility, and he has 32 publications. Now, someone once told me a while ago that, like I have, and I'm not boasting, I have like 700 publications, said if you can achieve 1 or 2 publications in the best journals of solid work in a year, that's as good as you can hope for. So Dan has 32 publications, 21 of them, 65%, were in Nature, Science, Cell, Neuron, or PNAS, which is like the top tier. So there's a lesson in there. Working in Newsom's lab, he started out in visual cortex, looking at the effects of visual perception on decision-making. But when he pursued his own line of investigation, he moved to the medial temporal lobe, and specifically the amygdala, and examined the effects of emotional processing of emotions and their effective emotional processing on cognition. And specifically, his lab is focused on the investigation of the neural basis of emotion and cognitive processes in these animal models. And the goal is to elucidate how neurons and neural circuits represent emotional parameters and how these representations relate to cognitive processes and how cognitive processes may regulate emotions. For those of you who know Dan, he sometimes seems like he can be a bit brusque and surly, but he's really a soft touch. And he's actually not just a pointy-headed scientist, he's pretty well-rounded, even Renaissance-like. If anybody wants to test that description, just ask him about wine sometime. So it's a pleasure to have him speaking to us today, and I'll turn it over to Dan Salzman.
Thank you, Jeff. It's too kind an introduction. I miss seeing everyone in person and the interactions we get in person. I know we all have been struggling with that really for almost 2 years now, without an end in sight. But I'm around, and I invite you all, to the extent that this talk thinks maybe we should be interacting more, even if it's over Zoom, just reach out to me and we'll get together, especially after the new year. It's always fun to come and talk about research, because that's what I do. And let me see, I'm going to now share the screen and go into the mode. And now I want to shrink that. I'm going to tell you a bit of a story about the lab, but most of the work that I'm going to tell you about, after what I would view as a somewhat long introduction, has to do with this paradigm shift in how we're thinking about studying how the brain represents information, mentalistic information, in order to produce different cognitive and emotional behavior. And the paradigm shift comes because in order to reveal the computational function of ensembles of neurons, our recent work is arguing that you have to examine what we call the geometry of how the brain represents information. And I think this is very important to psychiatry. And it's really a paradigm shift that we're arguing for, suggesting that we should no longer only care about what information the brain represents, but we need to consider how that information is represented. And really part of what I'm going to be doing in this talk is unpacking that for you. What do I mean about how versus what, and what exactly do I mean by geometry? And I invite you during the talk, I guess it's through Christine Denny, to ask questions seeking points of clarification, because the material is has a theoretical basis to it, and that's built on a collaboration, because the guts of the work I'm telling you about today is through collaboration with Stefano Fusi in the Center for Theoretical Neuroscience, and his colleagues such as Mattia Rigatti and Marcus Benna, who you can see listed here as key collaborators. And the guts of the experimental work and the main study I'm going to tell you about was actually work that Silvia Bernardi has done. And I mention all these names now because I always forget to mention the key people who really did all the work if I wait until the end, as well as the funding sources that supported either me, the work itself, or some of the people involved in the work. Okay, so let's move on. And in some ways, if you had to say, what did I do when I started to launch my own lab? I had done, as Jeff introduced, electrophysiological experiments in alert behaving monkeys to study neural mechanisms underlying visual perception. That's what I did as a student. And then when I started my own lab, I really made a very conscious decision to study brain mechanisms that I thought were going to be important for psychiatry, but still very focused on basic mechanisms. And these are some of the things that I want to try to understand in terms of brain mechanisms. And really the starting point of my lab had to do with thinking about how the brain represents variables that describe aspects of emotions. And in recent years, there's been recognition that that's not sufficient for understanding brain mechanisms that are important for psychiatry, because emotions don't operate alone. They interact heavily with and rely on different cognitive operations, such as conceptual learning, the ability to represent newly learned concepts, the ability to utilize concepts to what I'm going to explain in more detail later, I call generalization, which really has to do with the ability to use a concept that you've learned from past experiences to know what to do, think, or feel in a new situation quite quickly. And also, of course, reasoning and lots of executive functioning relies on our ability to form concepts. And as you will see later, somehow forming concepts runs the risk somehow of limiting cognitive flexibility of different sorts. And cognitive flexibility really has to do with adaptive abilities that are very important for mental health. And so one needs to understand the trade-off between learning concepts and limiting cognitive flexibility. And the argument that you're going to see play out in some of the talk has to do with the trade-off between conceptual learning and cognitive flexibility. And I'll be trying to explain that later. So these are some of the topics that my lab has been focused on over the years, not all of them. And there are other processes that are important for psychiatry. So there's been some work in the lab related to how emotion and attention interact in the brain. And I'm not touching on that today. I start off saying that we're really arguing for a paradigm shift. And I want to remind you in some ways what the traditional approach for studying these kinds of mechanisms has been. And in some ways, the approach would focus on examining the selectivity of individual neurons for variables in order to reveal their function in different cognitive or emotional operations. And this is often tested with electrophysiology, or nowadays, say, calcium imaging, to look at the response properties of cells in relation to a parameter. You might say this cell does X. And then you could also perturb activity in order to understand how the single neuron selectivity relates to those perturbations. And somehow the idea in traditional approaches is that the different functions mediated by different groups of neurons would be related to this single neuron selectivity. In fact, this is what my PhD thesis that Jeff briefly described was based on. But the new approach that really Silvia's work is championing is a different approach. It's an approach that emphasizes that in order to understand the computational function of a neural ensemble, the key thing to do is to examine the pattern of activity across the ensemble and across conditions. Now, in the real world, those are different environmental conditions, but in experiments, they're across different experimental conditions. And of course, to examine this pattern of activity across time as well. And overall, it's this pattern of activity across conditions that we refer to as the geometry of representations. And I'll explain this in more detail soon. But the notion is that this geometry, these patterns of activity, if you can develop different measures of the geometry, you can reveal the computational capability of an ensemble in ways that were previously not possible with the traditional approach, and in ways that may be critical for understanding how the geometry of neural representations helps you understand different aspects of the emotional functions of the brain, of the conceptual functions of the brain, of cognitive flexibility, and how these things interact with each other. So this in some ways is the theme. I want to plant all these seeds in your head. I'm not talking about psychiatric disorders. At the very end of the talk, I hope to give you a few examples of psychiatric disorders and how this analysis of the geometry representations may be informative. But this is in some ways the roadmap for the ideas that I want to go through in the talk. Now, in my lab, when I first started out, I made a decision to stay in the monkey preparation, but to go study things in that historically have been more studied in animal models and rodents, namely emotion and the function of the amygdala. So I was very much focused on number one on this list here. And rather than just trying to study this huge range of emotions, this is just an overly simplistic depiction of many different emotions, I basically took a more reductionistic approach and borrowed from what some people call the circumplex model of emotion, which says you can map all these different emotions onto orthogonal axes. Let me just switch to laser pointer mode. Orthogonal axes, one having to do with valence, going from unpleasant to pleasant, and the other having to do with intensity, going from low to high intensity, and with intensity somehow related to arousal. And this is, I didn't invent this model. Many people kind of use this framework for trying to say, you know what, we're going to simplify emotions in order to study mechanisms and think of them as being plotted in this two-dimensional space of valence or arousal. You obviously lose a lot when you're doing that, but in terms of going into animal systems, it can be quite powerful in order to simplify things in this way. And I was initially focused mostly on valence. We've done some work related to arousal that I'm not going to talk about today. And in our initial study in the amygdala, we really were going after the following, which was that most of the work in the amygdala had emphasized its role in aversive processing, especially fear conditioning and negative emotions. But was a general hypothesis out there in my head, which is the amygdala really just specialized for negative emotions? And so we went and did a study in the monkey where all monkeys had to do was a simple Pavlovian task, which a version of Pavlovian conditioning called trace conditioning. And trace conditioning is just one where there's a temporal gap called the trace interval that separates a CS from a US. And we did this with both appetitive Pavlovian conditioning and aversive, all built into the same task. So to perform the task, the monkey would first fixate, view an image. The image would be on the screen very briefly, about a third of a second. It would go off. There'd be a temporal gap of about a second and a half, and then you'd deliver an unconditioned stimulus. Now, one image, the images we used were novel in every experiment, and the animal had to learn associations between an image and either an appetitive or an aversive stimulus. So this particular image would be paired with a liquid reward. The monkey might see it first. Then on the next trial, we might see a different image. It was paired with an aversive air puff. Now, we used air puff as an aversive stimulus in the monkey because monkeys don't like it. In fact, humans don't like it either. You can actually do fear conditioning with air puff in humans, but it causes no tissue damage. So we can present it time and time again throughout an experiment in order to gain statistical power in looking at neural activity without doing any damage to the animal. And it's still aversive, and they'll take defensive behaviors and avoidant behaviors in order to avoid getting the full thrust of the air puff. So on the second trial, they might see an air puff, then maybe another air puff trial. We had a third stimulus type in the initial study that wasn't paired with anything. And this proceeded until the monkey learned these associations. And we knew the monkeys learned the associations because they developed two behaviors that would emerge during this trace interval. One is that they learned to lick in anticipation of reward, and the other is they used to blink, which is a defensive behavior in anticipation of an air puff gun pointed at their eyes. Okay, so once we knew that they had formed these associations between the stimuli and the appetitive and aversive events, we had a problem on our hands. We wanted to interpret the activity of individual neurons in the amygdala, but there was a confound built into the experiment if you ended it right now. Namely, if you saw a differential response to this image compared to this image, you would be left not knowing whether that differential response was due to the sensory properties of this image compared to this image, or was it due to the different reinforcement contingencies? And to tease those two things apart, what we did on the fly was a reversal in the contingencies. So the stimulus that was paired with something aversive became paired with something appetitive. The one that was paired with something appetitive became paired with something aversive. And the monkey learned those new associations on the fly. So by manipulating the reinforcement contingencies of the images, we're able to tease apart the responses related to the images compared to the responses related to expected appetitive or aversive reinforcement. And I'll just give you a couple of example neurons that from this study that was published, I guess, over 15 years ago now. So this was published back in 2006. On the left column is just example data from one neuron And this is example data in the right column from another neuron. We're plotting firing rate on the y-axis and the time from conditioned stimulus onset on the x-axis. This first dotted line is when the image comes on. The second dotted line is where the image goes off. And then there's this trace interval prior to the unconditioned stimulus. The unconditioned stimulus comes out here. And in this particular plot, we're not showing responses to it. So image 1 in this experiment was initially paired with reward. And if you looked at the cellular activity in response to that image, it went along firing at about 20 spikes per second. And then shortly after the image went off during this trace interval, you saw a very elevated response. Image 3 in the same experiment started off being paired with air puff, and it had a very low level of activity from the same neuron. Then you flip the contingencies. So image 1 is about to become aversive. Image 3 is about to become appetitive, and the cells flip their responses. And so we called this a positive cell because it responded more to an image when it was associated with something rewarding than something aversive, and it did so for both images, right? This is an example of the traditional approach that I was telling you about. This is all of a sudden making a claim that this is just a single cell that likes good things, right? By contrast, there were other cells that had the opposite valence preference. They preferred negative to positive, which was very reassuring, because if all you found in the amygdala were these sorts of cells, you would wonder what in the world is going on, because of the rich literature that already existed by this time showing the amygdala related to aversive processing. So here's a particular cell that has the opposite valence selectivity. For image 1, it responds more when image 1 is associated with something aversive, the air puff, than when the same image is associated with a reward, and it has the same relationship to valence for image 3. So this is what we call a negative cell. Now, one of the disadvantages of work in the monkey is that you don't have access to all the little tricks of molecular tools to label cells. You don't have all the tricks for being able to manipulate circuits. As best we could tell, these cells were anatomically intermingled in the amygdala. My PhD work had perturbed neural activity in a way related to physiological response properties and made predictions about the effect on behavior. But the success of those experiments was built on the fact that cells with similar physiological response properties were grouped together anatomically. So you could use a technique like stimulation and activate neurons that all coded for something very similar. Here, if you've got these positive and negative cells intermingled with each other and you start applying stimulation or lesioning or any pharmacological inactivation, you're going to hit both populations of cells, the positive and the negative cells. So you can't test any causal hypotheses about the relationship between the activity of these neurons and behavior because of that anatomical organization. And it was through discussions with Richard Axel that led me to do initially collaborative work in the rodent. Now we do some work on our own in the rodent to take advantage of some of the tricks that you can play in order to access neuronal populations in a mouse preparation and really extend this study from the monkey into the rodent. And again, this is still just following this track of trying to study how emotional variables are represented in the amygdala. And what we initially did is we developed a virus where we could express under the CFOS promoter something called channel rhodopsin, as well as a nuclear cherry. And also, channel rhodopsin was linked to a fluorescent protein. Now, CFOS is what's known as an early immediate gene, and it's expressed only when cells are activated. So in other words, we're going to express channel rhodopsin, and I think most of you probably have heard other people come through and talk about channel rhodopsin. This is this molecule that's been championed by Carl Diceroth and Ed Boyden and others that you can get into cells, and when it's expressed, if you flash light on the cell, you can activate the cell. If you use another type of channel, halorhodopsin or archirhodopsin, you can inhibit the cell. So it's a tool that's widely used in the study of neural circuits and animal model systems. So our goal, though, by linking the expression of channel rhodopsin to the expression of CFOS is only expressed when cells are activated. So we wanted to be able to somehow develop a strategy where we could selectively activate cells based on their physiological response properties. And this was a workaround in order to gain access to cells with different properties that were anatomically intermingled. Now, we had this nuclear cherry expressed because we needed to confirm that, in fact, we could label different populations of neurons with different physiological response properties that were in close anatomical proximity to each other. And there was a certain temporal dynamics to the expression of nuclear cherry and CFOS that we could take advantage of. So this particular vector not only got channel rhodopsin into the cell, but it also gave us what we refer to as a 2 time point marker of neuronal activity. CFOS is expressed, say, within an hour of activating a cell. If you look 1 hour after activating the cell to see if you have nuclear cherry expressed, it is not. It takes about 18 hours for it to build up. So that let us do the following type of experiment where we could inject a virus that got this expressed, wait 10 days until it's expressed in different neurons, then give a stimulus that will activate neurons. So here we go. We give the first stimulus, we inject the virus, we wait, we give a first stimulus. In this case, we're giving electric shock, wait 18 hours, and then give a second electric shock. If we stain for nuclear cherry, those are the cells that are activated by the first electric shock because it takes about 18 hours for nuclear cherry to build up. If we stain for CFOS, those are the cells activated by the second electric shock, not the first one, because CFOS comes and goes by the time 18 hours is over with. And then we can superimpose those two. And when you see, these are red cells, these are green cells. When you see yellow cells, there's overlap between the two. It's the same cells being expressed. And in fact, if you look at how many of the red cells were also labeled by CFOS, what's the overlap? It was a very, very high percentage here, about 85%. So it's a lot of overlap. By contrast, if we use two different appetitive stimuli, in this case, we're using nicotine at a dose that we knew was rewarding to the animal. If we give the virus, wait 10 days, give one nicotine dose, and then give another one 18 hours later, these are the cells activated by the first nicotine dose. These are the cells activated by the second nicotine dose. The yellow ones are the overlap. We get a high degree of overlap. Now, at this point in time, you don't know whether the nicotine activated cells and the shock activated cells are the same cells or different cells. You got to go do a different experiment. And here's that experiment done two ways. In this case, the first stimulus is a shock. The second one is a nicotine. So this red cells are shock sensitive cells. The green cells are nicotine sensitive cells. And now when you look here and you merge them, you see almost no yellow cells. So there's very, very little overlap between the two. And you can reverse the order, go nicotine and then shock. And again, you get very little overlap. And what you can see here is you do see red and green cells anatomically intermingled within each other, near each other in the basolateral amygdala. So this sort of confirmed the finding from the monkey electrophysiology that these so-called appetitive cells and aversive cells were anatomically intermingled. And it also said there's different populations for appetitiveness and aversiveness. And so that was pretty cool because not only could we label the cells this way, but these cells have channel rhodopsin expressed in it. So if we give a virus to an animal and then only give shock, we're only going to get channel rhodopsin expressed in shock sensitive cells. And then if we flash light, we can activate only the shock sensitive cells. And we can do the same thing by only activating the nicotine sensitive cells. And so we did those kinds of experiments measuring different parameters that were related to emotional states. So this is change in heart rate. So if we activate shock sensitive cells, we decrease heart rate. If we activate nicotine sensitive cells, we increase heart rate. This is respiratory rate. Again, we get valence specific effects of activating shock or nicotine sensitive cells. And if we activate shock sensitive cells, we can get the animal to freeze, which is a classic measure of being in an aversive state, but nicotine sensitive cells don't get you to freeze. And in fact, we could use the activation of these cells to actually instruct the animal to learn about a neutral condition stimulus. So if you activate shock sensitive cells, you can learn about a neutral condition stimulus and learn to avoid it. If you activate nicotine sensitive cells, you can use it as an unconditioned stimulus, and they can learn about a conditioned stimulus and actually learn to approach something that based on that conditioned stimulus. And so this was published in collaboration with Richard Axel about 6 years ago, and it really launched the mouse side of my lab that continues to try to take advantage of these different tools that you can use in the mouse to dissect neural circuitry. However, I don't really view this as being done. So this is very much in keeping with the initial goals of my lab, which was to study how different emotional functions work in the brain, initially in the monkey, and then taking a finding in the monkey and trying to test a causal hypothesis in the mouse. But we're really not done in terms of, I think, studying some of the key operations that must take place in order to account for normal mental health, but also that become dysfunctional in psychiatric disorders. So you could view in some ways what we've been doing in all the experiments I told you about so far is really looking at one-to-one mappings between stimuli and appetitive or aversive outcomes in behavior. And it's just not sufficient in terms of understanding brain mechanisms that are going to be important for psychiatry. And there's many reasons for that, but I want to take you through one line of reasoning that helps explain, in some ways, and motivate the work that I want to tell you about, the more recent work that I want to tell you about that was done by Silvia Bernardi in collaboration with Stefano Fusi and his group. And one of the major reasons is that you have to recognize that the same stimulus can produce very different emotions depending upon the situation. Now, that was true in the reversal learning task I told you about in the monkey. But what was happening there is the monkey was gradually relearning different mappings. And if that's what you had to do all the time, it's a problem because it's not very computationally efficient. It takes a long time to always relearn these different mappings. Moreover, not all situations are familiar. Many situations are quite novel. And in novel situations, you can imagine the learning task is going to be even greater. So novel situations and the ability to respond to stimuli rapidly in novel situations can be greatly enhanced if you can do something called generalization. So if you generalize from past experiences that might be somewhat similar to the current new one, then you can know what to do, think, or feel right away when you encounter a stimulus in that new situation. This generalization process relies on a process that we call abstraction. And abstraction is defined as the process of identifying features shared by different things, where these shared features essentially are describing the relational structure in the world. It might be a relational structure across time, space, or sensory or motor or internal states. Somehow it's telling you about the links between things in the world in an efficient way that lets you form what I would call a concept. These shared features that you can identify through abstraction would correspond to something like a conceptual understanding. And I'll unpack this more for you in the next few slides. Now, patients with psychiatric disorders often have difficulty not just with emotional processing, but with conceptual processing, right? Moreover, there is a price to pay if you only represent concepts and lose all the details of the world. You're going to impair your flexibility because you may not be able to respond in a way related to some of the details. And I'll explain that in more detail in a few minutes as well. So these are challenges that I think the traditional approach of looking at neural response properties has difficulty overcoming. Okay, so we have an internal environment. Somehow we generate concepts. And the first part of this talk is going to focus on what we mean by that. And concepts often can guide things like decision-making, action selection. They can be critical for emotion regulation because we adjust our emotions depending upon our conceptualization of situations, social situations, other things. And of course, concepts underlie learning, often memory formation, reasoning, and many other executive functions. In fact, learning one concept often helps you learn another concept that's related to it, et cetera. And if you lose concepts, or the ability to either form or utilize concepts, you can pair these different functions in different ways. And we really have a very limited understanding of how this actually happens in the brain. Okay, so I use the word abstraction as being a key thing, a key process that underlies the formation of concepts. So what do we mean by abstraction? Abstraction has to do with essentially coming up with what do you mean by abstract, right? What does it mean to abstract something? And what it means to abstract something, if you have two different things, you want to find the thing in common between those two things that are dissociated from any specific instance, right? So abstraction is the process of identifying shared features in a way that can generalize to other objects that might share that feature, but then you don't really, you can use it for any object that would share that feature, and you don't need all the different details of all the different instances in order to know that you have formed a concept describing it. Now, there's another meaning of the word abstract, which has to do with difficult to understand or abstruse. That's the one I encourage you to remind me with questions, if you're not understanding what I'm about to talk about, as I dive into what we think of as the right way for looking for the signature of abstraction in neural representations, because this is the part of the talk that's going to get a little bit more tricky. So the difficult to understand part is a cue for you to feel free to ask a question. And I'm not monitoring the questions, but Christine is. And if you have a question that's a point of clarification, just go ahead and ask it now or when it's happening. Okay. What's a concept? one way to view it is that you only retain the most relevant details. Here's two pens. Now, obviously, they have a lot of different features. In this case, they have colors, they have points, they have different things. But you can learn certain things about these pens to form different concepts. One object can be linked to multiple concepts, but one concept of a pen is that it writes. Another concept of a pen often is that the color of the pen is linked to the color it writes in. So you could learn that from just observing these two pens, that the pointy object is the part that points. This is a pen, it writes in the same color as this, et cetera. There's a concept or series of concepts you can learn about it. And the notion is only experiencing that with these blue and red pens, then all of a sudden, if you saw these pens of these other different colors, you would know right away that this pen's going to write in green, or that this pen's going to write in black, this pen's going to write in yellow, because you've generated this concept of a colored pen. In some ways, that's an example, a very simple example of a concept. Now, when we go to thinking about how concepts might be represented in the brain, this is what relates to the recent work that's going to be the focus of the rest of this talk. And this was published about a year ago in an incredibly dense paper. And my apologies for its density, but if you're have difficulty sleeping, you can use it to help treat your insomnia, and it will work wonders. In any event, first I want to emphasize, how do we study geometry of neural representations, and what, in order to reveal something about concepts or representations of concepts. I'm going to do this in a way divorced from the task that was studied in this paper, and just with an example from what's known as the MNIST data set. And the MNIST data set is this big set of digits, right? In point of fact, we're just going to use different instances of 1s, 2s, 3s, and 4s, and then also 5, 6, 7, 8s later. But in the paper, but here we're just going to focus on 1s, 2s, 3s, and 4s. And so you can learn the concept of 1s here. Each of these 1s is a little bit different. You can learn the concept of 2s. Each of them is a little different, 3s and 4s. But there are other concepts that you can form looking at these digits, right? You could learn the concept of 1s, 3s, but you can learn that 1s and 3s are odd and that 2s and 4s are even, okay? And those are concepts that you could learn. And so in principle, how would you actually think about how the brain might represent those things? Traditionally, if you were using a traditional approach, you would just look at neural responses to, say, odd numbers and even numbers in a single neuron and ask, how well can you classify odd and even? So imagine that we're only looking at the activity of neuron 1. In this case, we're plotting the activity of neuron 1 against the activity of neuron 2 in two conditions, a plus condition and a minus condition. If we think about it for a minute, the plus condition could be odds, this could be evens. But if you're only looking at the activity of neuron 1 and trying to classify odd from even in these two different conditions, you're essentially moving this green line along the x-axis, looking for the place where you can best separate the red and the blue data points. And you can't do too well. There's no place where you're going to correctly classify, get all the reds on one side and all the greens on the other side. And so this is a limitation that's present and here. And some of it is that the cells have complex firing patterns. But if you look at the activity of neuron 2 at the same time as you're looking at the activity of neuron 1, and use what's called the neural population decoder, which is something basically borrowed from machine learning that enables you to weight each neuron and to look for a threshold taking into account the activity of two neurons, you can find a plane, in this case a line because you're only looking in 2 dimensions, that completely separates the red and the green. And so this neural population decoder, if you take into account the activity of the pattern of activity in the two neurons in the two conditions, can classify odd from even perfectly well. And this kind of decoder is the main tool that's being used throughout all the data analysis that I'm going to talk about in the rest of the talk. Now, I just did it in 2 dimensions for simplicity, but in point of fact, when we look at neural activity recorded from an experimental subject, we have hundreds and hundreds of neurons. And I want to illustrate how different patterns of activity in neural ensembles, ultimately in an n-dimensional space where n equals the number of neurons, but at least in a higher dimensional space than 2, tells us something about the computational capability of the neural ensemble. And to illustrate how different geometries have different computational properties, I want to plot data not from 2 neurons, but instead from 3 neurons against each other in 4 conditions, okay? So this is a three-dimensional plot of neural activity. This is the firing rate of neuron 1, firing rate of neuron 2, firing rate of neuron 3, and we're going to have 4 different conditions for the 4 digits that I just told you about, 1s, 2s, 3s, and 4s. And in this case, I'm going to plot the points at random locations. Each of these points, you can view it as the average neural response to that condition. It's really a cloud of data points, and I'm just plotting the average of it. But that I'm choosing random locations in order to, the average location is random within the three-dimensional plot. So for instance, the average response to 1s might be here, and the average response to 2s here, 3s here, and 4s there. And if you put 4 random points in a three-dimensional space like this, it basically forms a tetrahedron, right? So the 1s and 3s here are connected by red, the 2s and 4s are connected by blue, these are odds, these are evens. If you play this game with a neural population decoder that I just told you about, the way the neural population decoder really works is something like the following, is that you take a subset of the 1s that were presented, a subset of the 4s, a subset of the 2s, and a subset of the 3s, and you train the decoder to find the plane that can best separate 1s and 3s from 2s and 4s. And then you test how well the decoder can do on reading out whether it's odd or even from trials not used from training the decoder. Now notice that when you're training the decoder here, you're training it on a subsample of all the different experimental conditions. And in this case, when you do that, if your average responses are in each of these 4 locations and your noise is not too big, you can find a hyperplane that separates the points just fine. This means you can classify odd from even really well, okay? Now, it turns out, because this is a relatively high dimensional representation, if instead of reading out odd from even, you wanted to read out small from large, separate the 1s, 2s from 3s, 4s, you could play the same game with the neural population decoder and find this plane here that separates the 1s and 2s from the 3s and 4s. And in point of fact, you could even find a plane that separates 1 and 4 from 2 and 3. That would be a plane that goes through like this. So what that means is that for this high dimensional representation, you can actually decode all the possible binary variables. You can decode 1s and 3s from 2s and 4s, that's parity, odd even. 1s and 2s from 3s and 4s, that's magnitude. And you can actually decode 1s and 4s and 2s and 3s. And if you just take the average decoding performance across all the possible binary variables, we're calling them dichotomies, it's 100%. And we defined a measure of geometry, which really has to do with the dimensionality of this representation. So we called it shattering dimensionality. It's how many ways you can shatter these 4 points, successfully and do the classification. So this is maximal shattering dimensionality in this particular representation. This has a particular power because when you have high shattering dimensionality, then you can do many possible readouts of this representation. So imagine I wanted to be able to read out odd from even. I can do it. If I wanted to be able to read out magnitude, I can do it. If I wanted to read out 1s and 4s versus 2s and 3s, I can do it. The ability to read out a representation of the world in flexible ways, in many different ways, that supports what I call cognitive flexibility. It's a key skill that we need to be able to have. If you just throw out tons of information, tons of detailed information so that these points overlapped, you wouldn't be able to do all these readouts and you would lose flexibility. So that's a pretty big price to pay, okay? But there's a price to pay for this high dimensional representation too. This particular geometry, I told you, plots the 4 points at random locations. If you plot 4 points at random locations, there's no way that there's built in any kind of relationship between the odd digits and the even or the even digits or the big digits and the small digits. There is no statistical relationship between them. They've just been plotted at different locations, at random locations. So what kind of geometry is going to give me the ability to capture a relationship between odd digits, for example, and even digits? Here's an example of a geometry that does that. In this particular geometry, if you look at the pattern of activity across the 3 neurons looking at 2s and 4s compared to 1s and 3s, they're clustered. So it responds in the same way, these neurons, to 2s and 4s, and it's a different way to 1s and 3s. This clustered geometry means that there's a hyperplane that separates the two for sure, but it also means that you've thrown out all the information. So now you can't separate magnitude, and you can't separate ones and fours from twos and threes. So your shattering dimensionality just went down because you've thrown out some information. But you have a new property here in its ability to generalize. And here's what I mean by generalize. If you take this decoder and instead of training it on all four conditions, you only train it on a subset of the conditions, here, ones and twos, you're going to find a hyperplane that separates the ones and twos. And it turns out it's going to separate the threes and fours correctly for odd and even just fine. And this ability to separate conditions that were not used to train to the decoder is equivalent to saying that the decoder can read out odd and even in a new situation. That type of generalization is how concepts are often used when we encounter a new situation. Okay, so this is really important, but it comes at a price, lower shattering dimensionality. So we're testing on threes and fours, having trained only on ones and two. We call this cross-condition generalization or cross-condition generalization performance. Now, if you look and try to apply that technique to this original geometry, if you train on ones and twos and come up with the hyperplane for it, you're going to misclassify threes and fours. So you can't generalize to novel conditions using this high dimensional representation. And one key question is are there geometries where you can find a compromise, where you can have high CCGP, support the generalization in new situations, and at the same time, still retain high enough dimensionality to do many readouts? Well, one geometry that we haven't considered is one where neurons allow you to represent more than one variable at a time in a way that supports generalization. Here, 4 points lie in a square in a plane, right? So I can train a decoder on ones and twos, find this hyperplane, and it'll classify odd even between 3 and 4 just fine. I can train a decoder to tell me about magnitude using ones and threes. It'll classify magnitude using twos and fours just fine. So now all of a sudden, I still have two variables represented, odd even and magnitude. But what I can't do is, and these are called factorized representations in the machine learning community. What I can't do, and these factorized representations are actually retained for any rotation of this planar geometry in the space. So now I've introduced mixed selectivity between neurons. So all the neurons now don't respond only to one variable. They respond to both magnitude and parity. So I've rotated this square, and the generalization properties for both odd even and magnitude are still there. But what you can't do in this perfectly plane representation is this third classification that we could originally do. You can't find a plane that's going to separate the mid-range ones from the extreme value ones. So this means that shattering dimensionality here is still not maximal, even though we have high generalizability for parity and magnitude. And now the key question is, how do we change the geometry that allows us to be able to achieve both things? And the way we do that is, and you can see it's possible, is just by perturbing the geometry a little bit. We've moved the fours and the ones a little bit. So now they no longer lie on a plane. It's a little bit like a square, but it's a perturbed square that's higher dimensional. So this still has all the generalization properties that I told you about. So you have high CCGP for two variables, but at the same time, you can find a plane that's going to separate twos and threes from ones and fours because you're back into a three-dimensional space, so long as things. So this is a compromised geometry. And in fact, there's a range of compromised geometry. We've played with this with simulations. There's a range of geometries where both CCGP and shattering dimensionality can be high. And the way you can think about that is you can think of this geometry here as being one potentially where you have this low dimensional scaffold that's embedded in a higher dimensional representation. And a key question we had going into the electrophysiological study done by Silvia Bernardi was whether this kind of geometry is achieved by the brain. And this, in our case, the abstract variable that we were interested in going in really had to do with situation or context, because the emotional meaning of stimuli, as we explained beforehand, can be very situation dependent. And if you think about it, Jeff mentioned my enthusiasm for wine. Here's one of my favorite wines. It's a bottle of Burgundy called Domaine de la Romanie Conti La Tâche. Here it's sitting at a picnic table. It's a picnic table I've never been to before, so it's a new environment for me. But because I've learned the concept of a dinner party, I know that this is a wine about to be served. Here's the same bottle sitting at a wine store. I've learned the concept of a wine store, even though I've never been to this one before. I know I have to pay a ton of money to get it. Here it's aversive. I'm in a sad state seeing it because I know I can't have it. Here I know I'm going to enjoy it. I can enter these environments right away and know what's going on because I can generalize my conceptual understanding of dinner parties and wine stores instantly and use it to interpret the emotional meaning of stimuli. We wanted a task that in some ways replicated some features of this, and we used a task that was similar to the reversal learning task I told you about before, now studied in monkeys, except instead of 2 stimuli that flipped contingencies, it's 4. And instead of having no operant contingencies in the old task, with the old tasks were just Pavlovian, this has operant contingencies. So there's 4 stimuli, A, B, C, D. The monkey initiates trials by holding a bar. For 2 of the stimuli, the correct response is to release the bar. That's A and C. For 2 of the stimuli, the correct response is to hold the bar. And then for 2 of the stimuli, a correct response gets you a reward. For 2, you get nothing, but if you get it wrong, you have to repeat the trial. So notice that the operant and operant contingencies are orthogonal to the reinforcement contingencies here. Then without warning, we switch the rules of the game. And so the animal goes from a context defined by this set of stimulus response outcome contingencies to a different set of stimulus outcome contingencies. So here the correct response was release, here it's hold, here it's release, here it's hold. We flipped around the operant contingencies, the reinforcement contingencies as well. And the monkey goes back and forth between these 2 contexts many, many times in these experiments. It's almost like we're trying to flip the meanings of the word la tâche and whether you should grab it or not grab it all throughout the experiment. Now, in order to solve this task, these context switches happen without warning. So the animal has to learn from an error because they don't know that they're switching from context 2 to context 1. And then the question is, do they then start performing trials correctly? And if they learn the concept of the temporal structure of the task, because there are certain sequences of trials that appear, here we are in context 1, here we are in context 2. An A release trial always follows, certain trial types. An A hold trial always follows different trial types. If you learn all these temporal sequences, or if you learn the inherent relationship between these 2 different contexts, you can actually know whether you should release or hold the bar based on your knowledge of the temporal statistics once you've got something wrong. And we know the monkey learns these temporal statistics because they use their knowledge of the temporal statistics to perform inference. So if you look at the last trial before one of these context switches, they're performing at a very high level, over 90% correct. The first trial after a context switch, they're performing poorly. And then on the very next trial, even though they haven't performed it in the next context, they're at above chance levels. It's a very next type of trial. So they haven't received A yet in this new context. And then for the Bs and the Ds, the first time they do it, they're performing even better. If you look at it, they're actually, once they start doing inference, this is what we would call inference because they're using their experience with trial C to know what to do on A, B, or D. They're actually asymptotic right away, just that they don't always perform inference on the first possible inference trial. Now, if you go into the neural representations, that we recorded in 3 brain areas in this study, hippocampus, dorsolateral prefrontal cortex, and ACC, you see a real hodgepodge of neural responses. These color codes show you how much responses you are. Yellows are more responses, blues are lower responses. Each column is a different condition, a different one of the 8 trial types in the experiment, 4 of them from context 1, 4 from context 2. Each row is a neuron. It's a hodgepodge of colors. These neurons, if you look at any given row, the cells have a complex pattern of firing. They're not firing to only one thing. So this is the kind of data set that makes you challenge the traditional view of neural coding, because it's hard to say what the cell simply does looking at this kind of pattern of activity. And it leads one to want to use these kinds of machine learning techniques. And indeed, if you use it, you can decode context very well in all 3 brain areas, looking at all of those cells. Here, the decoding accuracy is way above chance levels in all 3 brain areas. This is the size of the data set, how many neurons we're looking at. Now, these decodings aren't done in the 3-dimensional space. They're done in this n-dimensional space. Now, this is not telling us about the generalizability of the representation. This could be achieved with a geometry similar to that first geometry I showed you. Just all the data points are in random high-dimensional space. And in point of fact, there is no evidence that an abstract representation of context has actually emerged. The first hint that maybe something was there, we got by visualizing the data using a dimensionality reduction technique called multidimensional scaling. And I need to get the laser pointer off to show you. And we use that to essentially collapse the data into 3 dimensions and look at what the data looks like. And these 3 dimensions captured about 80% of the variance. And this is looking at the pattern of activity in context 1 and context 2 in the 4 different conditions. The rewarded conditions have this little extra yellow box around it. And when you rotate this is the data from the hippocampus. You can see this does not look like a random location. It almost looks like two somewhat perturbed planes that are opposite each other for the two different contexts. And if you look at the data from dorsolateral prefrontal cortex, it was kind of similar. These are these kind of a little bit perturbed planes, fairly parallel. It appears that there's some kind of geometric structure in the neural representation. When this is really just a visualization tool, but it told us, geez, we better develop measures of geometry that can capture the fact that these aren't just random representations of the different conditions. And the same was true in anterior cingulate cortex too, where things just rotate. So the measures we used in order to look at this were the two measures that I told you about already when we were looking at the MNIST data set. It's the cross-condition generalization performance, as well as shattering dimensionality. And what we did is we took an unbiased approach to look at all the possible dichotomies, all the possible ways of grouping these 8 conditions into 2 equal groups of 4. Now, some of these groupings have an easy name to it. Those are the context 1 and the context 2 conditions. Here's the rewarded and the unrewarded conditions. They're easy to name. But other ones of these dichotomies have difficult to name variables. Here it's A plus D versus C plus D, but you can imagine there's actually 35 different ways of doing it. The advantage of doing this is it lets us look in an unbiased way at all the possible information that the representation is representing and say, is there anything special about any of these dichotomies in terms of the geometry in which they're represented? And so when you go and do this, we were really interested in whether the geometry would look something like this, or something random, and that's what we took a look at. And so this is now a plot showing you decoding accuracy, or CCGP, on the y-axis for each of the 3 brain areas, where the open symbols are showing you decoding accuracy using what I would call traditional decoding, where you're training the decoder on a subset of the trials from all the conditions and testing it on held out conditions. This is like the plot of context I showed you. And this is cross-condition generalization performance. And what we've circled are 3 named dichotomies: context, value, and the operant action required. And what you can see is we're looking at a time interval right before the stimulus comes on. So this would be information about the prior trial. And that's still stored in memory right before the current trial. And you can imagine that when that stimulus comes on, in order to make a decision, you rapidly want to integrate it with information, especially about context, in order to know what to do. But if you look here, all the dichotomies can essentially be decoded, virtually all of them, in all 3 of the brain areas in this little time interval before a stimulus has even come on the screen. The best decoding is for context and value in hippocampus and the named dichotomies over here, but all of them are well above chance. This means shattering dimensionality is going to be relatively high. But if you look at CCGP, preferentially in the prefrontal areas, all 3 of these named variables are the ones that have this special geometry. In the hippocampus, it's just context and value, and it doesn't seem to have a special geometry for action. So this is a generalizable format for these named variables, but not the other variables. But at the same time, it looks like a representation where, because it's high shattering dimensionality, that it still retains the capability of doing many different readouts. Now, I'm not going to walk you through this particular analysis, but we did a very quantitative analysis to make sure that, in fact, a perfectly factorized representation that weren't perturbations like from the plane that I showed you earlier could account for the level of shattering dimensionality that we saw. And briefly, it wasn't. So this is a perfectly factorized representation would show you a shattering dimensionality that's around 0.6, but our observed shattering dimensionality was much higher. It was up at 0.7 in the hippocampus, and the same was true when we looked at the prefrontal areas. Now, the geometry of neural representations is not a fixed and static property. It's going to change as the animal engages the task. And in fact, that's exactly what happens here. So once an image comes on the screen, if you just try to decode the value of the current trial or the action of the current trial, you can imagine those are the 2 things the animal is trying to decide: what to do and what am I going to get. You can see within a few hundred milliseconds after an image comes on the screen, the animal is rapidly making a decision, because you can decode value and action very quickly within those few hundred milliseconds. And in fact, if you now go look at what happens to the geometry of representations, there's a big change, and we can discuss later or offline why there's a big change, especially when you look at how the geometry of the representation of context in the prefrontal areas. Because whereas in the earlier time interval, when we look back here, it was in this generalizable abstract format, now it's no longer in an abstract format, even though it's highly decodable still. So this representation still has high shattering dimensionality, but actually there's a loss in generalizability for the variable context. It's actually exactly what you would predict for brain areas implementing a decision on this task, but we can talk about that later. So high shattering dimensionality. All right, so what I've just shown you is that by considering the geometry of a neural representation, it's actually like a paradigm shift for thinking about how to get at the computational capability of a neural ensemble. It used to be that we just asked what information was represented, but that didn't tell you about whether the information was in a format that could be used to generalize. In the prefrontal cortex, the information about context here cannot be used to generalize because it's not in this abstract format. It doesn't have high CCGP, even though if you want to read out the information, it's there. So if you have to generalize in a new situation using that variable, you're not going to be able to do it using this representation when you see it there. If you look, however, using this approach, looking at cross-condition generalization performance, you can still find that multiple variables can be represented in an abstract format in all three brain areas and simultaneously, right? So this means that you don't have this pure clustering that I showed you in the first example of abstraction. You are able to represent information in a way that stores information about potentially multiple concepts, just like pens can have multiple concepts, so can these different situations. And it's this geometry, this abstract format reflecting abstraction that we would argue reflects the learning of concepts and can support generalization. These representations, however, don't discard all the detailed information about the brain because shattering dimensionality remains high in all brain areas. And this, we argue, is key for supporting flexible adjustments of behavior where you might need to make different responses by reading out the same neural representation in different ways, which is a key executive function that can also go awry in psychiatric disorders. The paper also has a modeling exercise showing that you get the same representations if you simulate the task using neural networks. So these may be general principles by which the brain constructs representations to be able to represent conceptual information at the same time as it's still retaining detailed information that supports flexibility. Okay, so let me just tell you briefly about the next steps with an eye towards psychiatry, because we really got into all of this going all the way back to the emotion work with an eye towards psychiatry. It's just a very long road. And Sylvia Stefano-Fusi and I have set up collaborations to move this work in a number of different directions. I'll tell you briefly about some of them here. But one key question is whether these neural representations, if you studied them in humans, exhibit the same principles where the geometry must be characterized to reveal the computational function. And to go after this, we've got collaborations with Daniel Kimmel and Daphne Shohami looking at fMRI, and we also have collaborations looking at single unit human electrophysiology in patients with Ralph Adolphs and Yuli Rodeshauser out in California, extending this work that was originally done in monkeys and humans, trying to document both using a similar type of measurement, single unit electrophysiology, but then extended into fMRI, whether these principles are actually going to be informative for studying a range of cognitive and emotional functions in humans. And if it is, that really sets the stage for us to do lots of interesting things in the future. A key question for us, and one thing that is somewhat lacking in the task that I just told you about, I didn't have a chance to go into all the details as to why, is we want to be able to link these measures more definitively with these two different distinct behaviors: generalization that relies on concepts and flexible responding that relies on shattering dimensionality. And we've got experiments being launched in humans as well as monkeys. That's a modified version of this task that sets up more conditions in which we can test the specific relationship between these measures of geometry and those behaviors. And then third, we really want to extend this study to try this analytic approach to study emotions in a new way, different than what we've studied before. Because I started off by saying there's a huge range of emotions, and then we use this kind of simplified model where we think of emotions as varying in intensity or valence. But in point of fact, emotions often have at least four properties: valence, intensity or arousal, the ability to persist over time, and in fact, emotions often generalize to new situations all the time as well. And so the question is, if you just focused a study in which you manipulated emotional state variables, whether again, these same principles of neurocoding would apply, where if you analyze the geometry, you can actually reveal how the same neural ensemble could confer all four of these properties all at the same time. You can imagine, for example, that one solution would be the brain has separate neural circuits for representing valence, for representing intensity or arousal. But an alternative hypothesis is that the same neural ensemble supports both valence and intensity or arousal, and just readouts of that representation that differ, that modulates outputs such as autonomic reactivity versus approach or defensive behavior, that accounts for why there's valence-specific behavioral effects and valence non-specific or arousal-related effects of representations of emotional states. And similarly, we're developing new tools to look at slower timescales, to look at persistence, and trying to develop tasks where we can study generalizability of emotional states from one situation to another. So this is an area that we're actually going to try to pursue in both mice and monkeys. It's for, I think, somewhat obvious reasons. Trying to study these very abstract contexts or situations in mice are quite challenging, but I think we can possibly gain significant leverage in mice to go after these emotional state variables. Now, this is our next steps, but we really have an eye, if we can achieve some of those things, to move on these studies in a collaborative fashion to study clinical populations. I'm just going to give you a few examples of that. These are not necessarily even terribly well thought through, and they're not necessarily the first ones we would go to, but it's pretty obvious that if you start thinking of dysfunction in clinical populations, you can see that many different clinical symptoms can be thought of in terms of failures to generalize. So this could be due to a deficit in conceptual learning or in using neural representations, conceptual information to be able to generalize. And, you know, if you think about schizophrenia, patients with schizophrenia often exhibit a lot of concrete thinking, which is sort of the opposite of abstraction. So if you fail to generalize accurately, in part due perhaps to generate these conceptual understandings of environmental information to assess threats, et cetera, it's easy to see how that could lead to paranoia. It would be interesting to study, you'd have to design somewhat different tasks than we have to, but to study the geometry of neural representations in the context of such situations in schizophrenia and see if these principles of coding that look at geometry would be informative in understanding differential processing in a totally different way than people have looked at previously. Similarly, you can think of other psychiatric disorders, PTSD or borderline personality disorders. These are disorders with often abrupt dysregulation of emotion, yet quite prominent dysregulation of emotion. And again, this can be triggered with an inability to generalize and an understanding of a social or environmental situation to a new circumstance that triggers a very strong emotional response that's maladaptive. So, one could pull out other psychiatric disorders and also look at either deficits in conceptual learning or utilization in some very interesting way. But, failures in flexible behavior, of course, which would be related to having a high dimensional representation, could also be examined in a different way than is done previously. For example, patients with schizophrenia that are sometimes described to have difficulty with tasks that demand high, cognitive loads and working memory. This may be correlated with actually decreased shattering dimensionality in prefrontal neural representations. And that may be the reflection of the difficulty of storing all the information needed in order to exhibit the type of flexibility it is to perform some of those types of tasks. So this isn't necessarily the first patients we would go to. I think we need to validate these tools in human models first, just using regular patients. But these tools are a new way of thinking about how to look at the brain represents information, and therefore a new way of thinking about what goes awry in different psychiatric disorders, and potentially an assay for when you can actually alleviate symptoms and what would be the neural signature of that. Okay, I'm going to stop and take questions. I got no interruptions, so I assume I was crystal clear or everyone was very shy. But I want to thank all my collaborators and supporters, and thank you all for your attention. And Christine, I think you now direct me as to what question to answer. Is that right?
Yes. First, I'm going to start off by reminding the attendees that if they are a trainee, a resident, a postdoc, or a grad student, to please write trainee before your question, and we're going to give priority to those questions first. Dr. Lieberman, I think, wants to ask 2 questions first, and so I'll let him ask them if he doesn't want me to read them, and then I can start asking other questions.
Okay. Dan, that was an absolutely brilliant talk, very interesting. Maybe I missed it, but the pattern of activity I described at the beginning that forms the geometry, what are the parameters? Is it spatial, temporal, magnitude, or what?
Well, when we're talking about the pattern of activity, you can choose a time window. You can choose whatever time window you want. We tend to choose relatively short time windows with electrophysiological data because we have good temporal resolution. With fMRI data, where you don't have as good temporal resolution, you may not be able to play the game quite the same way. But you choose a given time interval, and you just count how many action potentials happen in that time window in each neuron. and then you plot it in this firing, what I would call firing rate space. So you plot the activity of each neuron against each other in that n-dimensional space. If it's 3 neurons, it's a 3-dimensional space. For each trial, if it's an experiment, in each experimental condition. That's the geometry. It's just the activity in a defined temporal window for each condition, or each trial and each condition. Now, you can slide that window in time, and what you have is an evolving geometry. And I showed you data from 2 different 900 millisecond time windows, basically. One right before an image came on the screen in the task, and one after the image came on the screen in the task. In point of fact, we've used shorter time intervals for this, and we've used longer time intervals for it. So you can choose your time interval, and depending upon the number of events you have, these analyses can work well or not as well. When you, for example, when you're using calcium imaging data, you don't get as many events as you do when you're doing electrophysiology. And so you might tend to use bigger windows, for example, to have more events. But basically it's the pattern of activity at in a given time window across neurons and across conditions plotted in this high dimensional space. That's the geometry.
How many cells can you record from simultaneously?
So we can record from more and more with new technology, but in most electrophysiology experiments, and also in a lot of calcium imaging experiments, people make this assumption of pseudo-simultaneity, whereby you combine the data across different experimental sessions in a way that where you are destroying the correlations between neurons recorded simultaneously when you do that, but you can still look at the geometry by, if you have condition 1 from experiment 1, you can take condition 1 from experimental 2 and put the data together, essentially. And so all the analyses in monkey, and this is true of almost every monkey study there is, use this kind of pseudo-simultaneous analysis. When your channel count gets high enough so that your yield is really high, really, really high, like if you were getting hundreds of neurons simultaneously in a given brain area, you probably could move away from that and study the effective correlations by using simultaneous recording. And with fMRI, you can potentially do that, although you're more limited in terms of your temporal resolution, and you're often more limited in terms of the number of trials you have. So there's a trade-off across the techniques that makes them complementary and makes it challenging. So it's not entirely straightforward just to do exactly what we've been doing and just map it into fMRI. It's challenging.
And finally, just in terms of your extrapolation to mental disorders, I would say that schizophrenia might be more representative, at least at the early stage of the illness, of the cognitive flexibility and being over-inclusive, because things get to, and Guillermo Horga and Gaurav Patel have been working on this. They tend to, just incorporate stimuli and environmental elements into their conceptual thinking, which is oftentimes delusional. The other thing is that in terms of the restricted, autism and OCD would seem to be exemplars.
Yeah, no, I agree with that. I didn't want to just go through all the psychiatric disorders. I think one of the things that would be interesting is, to the extent that these measures can first be validated in humans, period, that would be great. But if they can be, then you can move them into patient populations, and they can be studied as a function of the course of illness in patient populations as well. And that might be, and I think that's what you're suggesting for schizophrenia, for example, that there may be an evolution even in what the geometry looks like as the disease evolves. And I was suggesting towards the end that it could be that some forms of effective treatment for some types of disorders could also be reflected by changes in the geometry representation of function of treatment. I mean, this is all fairly speculative, but the way my basic research works is that I always have an eye out for trying to find out things that I think, at some point in time, and the time scale is slow because progress is slow, there'll be a kernel that would be useful. And this is sort of the kernel of this type of work.
Thank you. Okay, Dr. Lehman, if you're done, we have a bunch of trainee questions, which I'm going to prioritize first for you, Dan. Andrew Chen said, really cool talk, a couple of questions. While the representations in your presentation are 3D, I imagine that the actual original neuronal activity space is very high dimensional. Do these metrics such as CCG be also perform well in very high dimensional space? Or is it the typical workflow to use dimensionality reduction techniques first? So that's the first part of the question I'll ask.
Yeah, it's a great question. And, this, in some ways, I packed a lot into the talk, and so I didn't emphasize this point enough. When I explained how we do CCGP initially and how we calculate shattering dimensionality, I was doing it in a 3-dimensional space. And then, of course, when I showed those multidimensional scaling plots, that's a 3-dimensional space as well. The first part was just used to explain how the techniques are done. And the multidimensional scaling plots are just a visualization technique. All the analyses are actually done in a very high dimensional space where the number of dimensions equals the number of neurons studied. So for example, in the hippocampus, there was about 650 neurons. So CCGP and shattering dimensionality were calculated in a 650 dimensional space. So it's always applied in the full dimensional space. Now, we sometimes, if we want to compare two brain areas where we have different numbers of neurons, we don't want to compare the values, we're either downsample or upsample so that it's a fair game. So we're comparing equal numbers of neurons. But the analyses are built to be able to study representations in a high dimensional space where the number of dimensions is defined by the number of neurons. If you're talking about single neuron electrophysiology, if you're talking about fMRI, then you're talking about a neuron is like a voxel. And so that's also going to be high dimensional because fMRI grabs lots of voxels simultaneously. And that's what I was referring to earlier with regard to Jeff's question when I said with fMRI, because you capture data from so many voxels all at once, you don't necessarily want to play the pseudo-simultaneous game that we play with electrophysiological data. That's the first part, right?
That's the first part of the question. In the second part, in the first set of graphs you showed with decoding accuracy over time, how is decoding accuracy so high, 70 to 80%, prior to the onset of the stimulus?
Yeah, it's high because you have enough of a memory trace of what has been happening from the last trial and maybe even trials before it, because we can decode information about things 2 trial back. So the brain is not just simply forgetting just what happened. It has a memory trace of what's just happened. What the form of that memory trace is what we think is being captured by the geometry. Some of the information about past trials is represented in what we call an abstract format, so it can be used for generalization. Some of it is not. But in point of fact, you still have information about what recently happened during that time. And some of that information is incredibly useful for what you're about to do when the image comes on the screen in this simple task. So it is true that that decoding performance is quite high then, but we've seen that before on even simpler tasks. And other people have seen traces of activity during intertrial intervals before that tell you about the recent past. There have been papers going back probably more than 30 years showing you signals during these so-called intertrial intervals that aren't pure noise, but are instead telling you about recent events.
Thank you. We have another trainee question, 2 questions, but much shorter. How do you know that the measures of geometry you've developed are the most useful measures? How does your work relate to the dynamical system approaches used in other groups to characterize geometry?
Well, that's a big question. So I'm not sure I'm going to do a great job answering that. Our approach, a lot of groups that are measuring geometry are using dimensionality reduction techniques and presenting that as a way of capturing what's going on and then looking at the dynamics of it. I'm not so aware of another paper that makes this formal proposal that the way to think about how a concept is represented is by creating tasks where you can train a decoder on some conditions and read out a decoder on conditions that were not used for training. So they're novel conditions to the decoder. I'm not sure I know of a analog measure where that kind of formal definition of what it means to represent a variable in a way reflecting abstraction is out there. But without knowing exactly which measures, a lot of time when people talk about the dimensionality of a neural representation, they're using a type of measure of dimensionality called principal component, based on principal component analysis. And without going into all the details, that measure of dimensionality is definitely different from what we call shattering dimensionality. Shattering dimensionality can be correlated with dimensionality measures using principal component approaches, but it can be not correlated with it as well. Because we really are interested in using this measure of dimensionality to tell us about how many input-output operations can the representation support, which we think is important for flexibility. The principal component analysis approach doesn't necessarily give you that very handily. And there's a supplementary figure in Sylvia's cell paper that actually shows you the principal component-based analysis and how it modulates in time. So you can compare it to the shattering dimensionality and see how it differs and is the same. And in fact, there's a part of the discussion section that discusses that as well. But it gets quite nuanced. And we actually move into the arena where, if Fusi and I sometimes have given talks back to back on this work, and we trade questions back and forth, Stefano would probably have more to say about this topic than me. But without knowing specifically which other measures you have in mind, I'm just not aware of another paper that makes this very specific proposal about how do you represent abstract concepts, and moreover, how can you think about a representational geometry that still retains information that lets you support flexible behavior at the same time. I'm not aware of work that really has taken that slant on things.
Thank you. There's a question from Alex Harris, elegant work. Are there ways of artificially increasing the shattering dimensionality with, for example, optogenetic interventions?
Yeah, so I think one of the challenging things about this new paradigm is it's not so clear how to use optogenetics or any perturbation experiment in order to test hypotheses, because what you do when you perturb, and many of you don't know this, I did my PhD thesis doing perturbation experiments using electrical microstimulation, and obviously I showed you in the mouse we do optogenetics, and we've done chemogenetics too. What you do with these artificial perturbations is you essentially move the pattern of activity that's in a neural ensemble into a state that it probably never achieves normally, because you're controlling it and making the pattern different than is normally realized. You can try and do things like we did in the mouse work that I told you about briefly, where we activate cells that only respond to one particular thing. But in all honesty, when we do activate those cells, we're not replicating a state that probably is normally realized in the brain. So now, can we randomly activate neurons all over the place in a way that could increase shattering dimensionality? It's quite possible we could. I will say that shattering dimensionality is often quite high in prefrontal areas in general, and Stefano Fusi has done previous work showing this in other papers as well. So detecting the big increases through optogenetics may be challenging. So you could probably do it, but when you do that, you're probably moving to a spot in what people refer to as the neural manifold. It's a spot in the firing rate space that's not normally realized. So interpreting what that means may be a little bit challenging. So it's sometimes hard to develop experiments, thoughtful experiments where you're perturbing activity to test some of the main hypotheses put forth by this geometry idea. That's why our first step is to develop tasks that we think have lots of conditions that let us look for neural correlations. And this is not unlike what people first did when they used traditional approaches. I mean, if you think back to some of the early work, people would look for neural correlates of a perceptual decision, where they're looking for how the responses to manipulations of a particular stimulus variable cause changes in neural activity that's related to perceptual decisions. That's all correlative work. And then sometimes, and there aren't actually that many examples of it, people have been able to go do perturbation experiments to show that actually the correlation is predictive of causality. So I think it's a challenging problem. We have some experiments that we have in mind that involve looking at functional interactions between brain areas in the mouse by inactivating one brain area and looking at how it affects geometry in another brain area to see if we can understand how brain areas functionally interact to produce certain geometries. And the way we've thought about that more is when we're getting ready to go after these emotional state variables that I talked about at the end.
It is 12:30, but if you're open to staying, we have so many questions. I don't know if you're
Okay.
open to meeting.
I'm fine. I'm
I
fine.
can read you more
And I'm
questions.
still energetic. I'm still energetic. So
We still have over 100 attendees, so I'm going to keep reading for a while, and you tell us when you're done.
no, I have a meeting for another hour and a half.
Okay, I
I'll
can't
be the
read
last
together
one
in our house. Okay, Jeff Miller has a question, and he says, "Thanks for this amazing talk. Can you speak about the relationship between the geometry you are speaking about and the anatomy? The planes you draw are in hyperspace. Are neurons that are close to one another in this machine learning hyperspace close to one another in the atomic space or anatomic space? Or if not, how would you identify commonalities between neurons on one side of the hyperplane?"
Yeah, so I will say, I mean, I think that's ultimately a real mechanistic understanding of geometry ideally will map things back into circuits. And this is one of the reasons that we've been interested in going into the mouse and looking at geometry. So the experiments that we proposed looking at mouse, for example, we're going to use 2-photon imaging where we can actually see the cells, put our fingers on them, so to speak, even know something about the cells, maybe about their projection targets or cell types, et cetera. So, we have in mind quite a range of things exploiting what's available in the mouse to be able to look then more precisely about these things. In the monkey, I think that's an extraordinarily challenging problem. If you were going in with an array into a cortical surface area, you might be able to do something because now you kind of are spanning a whole piece of tissue. When you're in these deep brain structures, and across, you're advancing electrodes in the brain many times in different spots, and then using pseudo simultaneous recording. I'm quite humble about the possibilities for gaining the type of anatomical insight you're seeking from the monkey on geometry. It may be challenging. I think the real opportunity here for this kind of thing may exist somewhat in fMRI in a coarser way, because you can measure all these different voxels all at the same time. You get it, right? You can get an entire, all of the hippocampus. You can get it once, and you could subdivide a given brain structure into different subregions and take the voxels in the anterior part, the voxels in the post. You can do all those things. The caveat is that one voxel has a lot of neurons in it, and so you may be missing things that are actually quite informative. So I think it's a hard question to get at, how to map the geometry into more precise anatomical locations. In all the studies that I presented and that we've done, it's many experiments to get those. It's about 2,000 neurons in that paper. I forget how many experiments. It might be 50 experiments to get 40, 50 experiments to get all of that data, and then it's combined in a pseudo-simultaneous way. We can reconstruct where all the recordings come from, but being able to link those locations back into the geometry, I don't think that's realistic with that style of experiments. We need better techniques. So I would hold out the most hope for the mouse, because we get cellular level resolution. And for the targeted brain area, we may actually get quite a bit of information about this. fMRI, I would hold out some hope for as well, but that it's possible that the loss of spatial resolution or temporal resolution, or both, could be costly there. It's too early to tell.
Okay, so I think you answered actually some other questions in that question, which I'm thrilled about. There is a question from an anonymous attendee. Is there any reason to think that the neuronal noise in representation contributes to greater generalization or CCGB?
Yeah, I mean, if you have too much noise, you're going to impair both traditional decoding and CCGP without a doubt. And so, I mean, that's certainly true. And, we get with single unit electrophysiology, or you get measures, you get repeated measures. So you have a sense of the noise that you're dealing with. And, we can also do simulations and look at how the noise affects things. So if you have too much noise, if you listened very carefully when I was first explaining decoding, I usually have a sentence in that says something like, okay, so I'm plotting these points in this firing space at these 4 random locations. And if the noise is not too large, then you can find a hyperplane that separates these 2 points from that point. And that's always going to be true. It's always the average response could be different, but the noise could be so big that the decoding, whether it's traditional decoding or CCGP, could fall apart. So that's always going to be true. And that could mean one of two things, or it could mean one of many things, but sometimes that's a sample size issue. Other times it could be that you're just in the wrong brain area. And if you went to a different brain area, you would find things aren't so noisy. And if the animal's just using that other brain area that you're not studying, you would find a much cleaner signal, so to speak.
We have a question from Mike Schmidt. What a beautiful and powerful set of ideas. Thank you. I listened to most of this talk with Jeff Hawkins' Thousand Brains ideas in mind. Have you considered investigating how this geometry can be assessed in the cortex, whether it is mapped intracolumn or across columns, or if cortical columns represent two layers of organization?
Yeah, so I mean, obviously you would want to go to brain areas where you have a pretty clear columnar organization. And that's not true of hippocampus. I'm not even really sure that I could say that I'm aware that it's really true in DLPFC or ACC. One of the things for years, people would study parts of the prefrontal cortex, and almost no matter what the study was in monkey electrophysiology, for example, and honestly, it's true often in human fMRI, you would wind up with this, in a monkey electrophysiology study, you would wind up with, you design some complex cognitive task, and you wind up, the paper reports that 23% of neurons encoded this variable that's needed for the task. And almost no matter what question you asked, it turned out the prefrontal cortex, it could be used to decode, you could find the variable represented. And then people like Stefano Fusi started pointing out that actually prefrontal neurons don't exhibit what's called pure selectivity. They exhibit what's called mixed selectivity. And mixed selectivity, I talked about this briefly in the early part of the talk, means that cells are going to respond to some either linear or nonlinear combination of different variables. And as soon as you have populations of mixed selectivity neurons, then it becomes more and more complicated to even define what columnar organization would be. If you go back to early sensory areas, you have much more clear-cut columnar organization, but even there may be more mixed selectivity than people have realized, because a lot of the early experiments would only manipulate one parameter at a time when they're in a given study. It would be interesting to go back, and I think some people are starting to do this, say in the visual system, to go back into visual areas to be able to look at dimensionality of neural representations, especially people interested in object recognition are using approaches that in some ways have a tight relationship to some of our work on abstraction. And so, sometimes people say, am I interested in going back? Because, when we're looking at the hippocampus, we could easily just go back to infratemporal cortex or even earlier visual cortex and look at what point do you not see abstract representations of context. So I think it would be interesting to go look at this as a function of columns, although I don't necessarily think that this is going to be, geometries revealed only, when you go back to earlier visual cortex, as an example, the receptive fields get quite small. And so depending upon the nature of your task, you may not be able to look at this in an interesting way in individual columns.
Okay, we have a question from Guillermo. Amazing work and talk. In translating this to clinical populations with clear cognitive deficits, you may see patients who cannot learn this type of complex reversal learning task well. How could you tell if they cannot learn because of an issue of representational geometry or vice versa? They never develop this geometry because they don't learn the task variables to begin with?
Yeah, well, two points. One is that I'm not 100% sure that we would want to use this exact task, although we are using a very close analog to it in human fMRI studies, as well as the human electrophysiology studies that I talked about being done collaboratively. And I will say that, so the human electrophysiology studies, of course, are done in patient populations. These are patients with epilepsy. And there's considerable individual variability in how well patients perform the task. And one of the interesting things, one of the signatures that we use to say that they know something about the task has to do with this inference behavior that I showed you that the monkeys have, and humans illustrate the same inference behavior when they do the task well. Some human patients with implanted electrodes don't seem to get it. They don't seem to show inference. However, if you halfway through the experiment, you see they're not getting it, often you know that they're not going to get it. And if you interrupt the experiment briefly and then explain the task to them and say, look, you're switching back and forth between two contexts where the two contexts are defined by the set of stimulus response outcome mappings, it's an aha moment for them. And you can then, after that verbal instruction, watch them all of a sudden demonstrate inference when they perform the task. Now, I don't know if the same thing would work in a patient with schizophrenia or some other psychiatric disorder, but there is the possibility in humans to use language in ways that you cannot in animal models. So one simple thing that might be worth trying in a patient population that's not learning the task is to see whether the verbal instruction gets them to learn the task. And then you can examine the geometry before and after such instruction to see if it matters. I mean, that's my first idea. My second idea is to design tasks that can ask some of the same questions that might be simpler and easier to grasp. So probably one would try both in parallel.
Thank you. We have another trainee question now. Your experiments seem to rely on tasks with many reversals. Do you have any idea how switching activity in basal ganglia relates to these limbic and cortical areas and their geometry?
No.
Okay.
No,
Okay.
we haven't studied basal ganglia. I think the basal ganglia is going to kick in, especially when you use really well-trained stimuli. Most recently, when we're doing these tasks, we're using new stimuli more often. And and it's not that the basal ganglia won't be involved, but the more habitual you make the context switches, the more I expect the basal ganglia to kick in, but we just haven't been there.
Last question, I promise. This is a great set of tools that you and your collaborators have created. My question is about the stability of the neural representation in an individual. If you, for instance, change the visual stimuli in the task, does the activity pattern across neurons for each of the conditions change, or are they invariant to the change in the visual stimuli?
I mean, that's a great question. So the published paper used the same set of images that were well learned. We have data from some new subjects and some new experiments going on that are swapping out either subsets of images or all the images that would let us look at this systematically, but we just haven't done it yet. But it's a great question, and it's a question that's in our mind about, you know, if you create the subspace for context one, but then swap in new images, are you using the same subspace kind of thing? And so we have those sorts of questions in our mind and are going after them.
I have questions for you, but I'm going to email you. So we had a break, and it's mostly about publicity stuff. So I just want to thank you from everybody up here for taking the time to give the talk today and staying extra and answering all of these questions. It's been a real pleasure, and it's great to see you on Zoom. And I'll see you.
No, it's a lot of fun. I'm so sorry I couldn't be in person, but hopefully the next time I give grand rounds will be in person again. So
that would be wonderful. Thank you so much again, and I hope you have a great day.
Okay, bye-bye.